Hi everyone, myself Aishwarya Sibalan, first MSc Biology, St. Mary's College, Thrissur. The topic is Cellular Components of the Immune System, Lymphocytes and Phagocytes. Lymphocytes. Lymphocytes are the central cells of the immune system responsible for adaptive immunity and the immunological attributes of diversity, specificity, memory, and self-non-self recognition. The other type of white blood cells play important roles engulfing and destroying microorganisms, presenting antigens and secreting cytokines. Lymphocytes contributes 20 to 40% of the body white blood cells and 99% of the cells in the lymph. Lymphocytes can be broadly subdivided into three populations: B cells, T cells, natural killer cells on the basis of function and cell membrane components. Natural killer cells or NK cells are large granular lymphocytes that do not express a set of surface markers typical of B or T cells. B and T lymphocytes are small, motile, non-phagocytic cells which cannot be distinguished morphologically. B and T lymphocytes that have not interacted with the antigen, referred to as naive or unprimed, are resting cells in the G0 phase of the cell cycle known as small lymphocytes. Interaction of small lymphocytes with the antigen, they progress through the cell cycle and lymphocytes enlarge into blast cells called lymphoblasts. Lymphoblasts proliferate and eventually differentiate into effector cells or into memory cells. Effector cells function in various ways to eliminate antigen. Plasma cells, the antibody secreting effector cells of the B cell lineage, have a characteristic cytoplasm that contains abundant reticulum and also many Golgi apparatus. B lymphocytes. The B lymphocytes derive its letter designation from its site of maturation in the bursa of Fabricius in birds. For bone marrow is its major site of maturation in a number of mammalian species, including humans and mice. Mature B cells distinguished from other lymphocytes by their synthesis and display of membrane-bound immunoglobulin molecules, which serve as receptors for antigen. Among the other molecules expressed on the membrane of mature B cells are B220 is frequently used as a marker for B cells and their precursors. CR1 and CR2 are receptors for certain complement products. CD35 is a receptor for IgG, a type of antibody. Class 2 MHC molecules permit the B cell to function as an antigen present in cells APC. T lymphocytes. T lymphocytes derive their name from their site of maturation in thymus. Antigen binding T cell receptor is structurally distinct from immunoglobulin. It does share some common structural features with the immunoglobulin molecules, most notably in the structure of its antigen binding sites. Most T cells recognize antigen only when it is bound to a self molecule encoded by genes within the major histocompatibility complex. Most mature T cells express the membrane molecules are CD28, a receptor for the co-stimulatory B7 family of molecules present on B cells and other antigen present in cells. CD45 is a transduction molecule. Natural killer cells. Natural killer cells was first described in 1976 when it was shown that the body contains a small population of large granular lymphocytes that it displays cytotoxic activity against a wide range of tumor cells in the absence of any previous immunization with the tumor. NK cells plays an important role in host defense both against tumor cells and against self-infected with some, though not all, viruses. These cells, which constitute 5 to 10% of lymphocytes in human peripheral blood, do not express the membrane molecules and receptors that distinguish T and B cell lineage. NK cells possess spontaneous cytotoxicity towards various target cells, meaning malignant and virus-infected cells. Their cytotoxicity is not antibody dependent or MHC restricted. Their activity is natural or non-immune as it does not require sensitization by prior antigenic contacts. Phagocytosis. Macrophages are capable of ingesting and digesting exogenous antigens such as whole microorganisms and insoluble particles and endogenous matter such as injured or dead host cells, cellular debris, and activated protein factors. In the first step in phagocytosis, macrophages are attracted by and move towards the variety of substances generated in an immune response. This process called chemotaxis. The next step in phagocytosis is adherence of the antigen to the macrophage cell membrane. The macrophage membrane has receptor for certain class of antibody. If an antigen is coated with the appropriate antibody receptor on the macrophage membrane more readily than antigen alone, and phagocytosis is enhanced. Thus, an antibody function as an opsonin a molecule that binds to both the antigen and the macrophages and enhance phagocytosis. Granulocytic cells. The granulocytes are classified as neutrophils, eosinophils, or basophils. The neutrophils has a multi-node nucleus and a granulated cytoplasm that stains with both the acidic and the basic dyes. It is often called a polymorphonuclear leukocytes. The eosinophils has a bilobed nucleus and a granulated cytoplasm that stains with the acidic dye is in red. The basophil has a lobed nucleus and a heavily granulated cytoplasm that stains with the basic dye methylene blue. Both the neutrophils and eosinophils are phagocytes, whereas basophils are not. Neutrophils, which constitute 15 to 70% of the circulating white blood cells, are much more numerous than eosinophils or basophils. Thank you.